So I'd like to start with just introducing everyone to Terumo BCT. We are a diversified company, so the cell therapy technologies business is one of 4 businesses within the company. And really our history and heritage is in automation. So automating complex technologies and processes, whether it's in a blood center environment, in a hospital, or in a complex manufacturing environment, which is really where our cell therapy technologies business resides. And that's where I'm going to focus the discussion today. And as we work with different customers in the space as a tool provider, so we are not developing a therapy, which might make us unique in this session, but we are tool providers enabling our customers to be able to bring these life-changing therapies to market. And as we engage with them and really understand what do they need in order to be able to advance their therapies, there's some key themes that keep coming up. So reproducibility, quality, and quality of the final product, but also quality throughout the entire process and all the components that feed that process. GMP compliance as they move towards commercial manufacturing, and ability to scale, particularly for those allogeneic processes. And our customers all acknowledge that, automation is needed. It's not a question of do we automate, but rather it's a matter of when's the right time to automate, and what are the processes that can be automated, and once you have complex process, how do you integrate automation into that process? And what's interesting about automation is, if you look at the continuum of development from preclinical to commercial, and you look at manual processes and fully automated, there's a linear relationship. However, if you wait until you have a commercialized therapy to automate, your switching costs are very high. And a lot of our commercial leaders and those who have led the frontier in cell and gene therapy have discovered this. And so a lesson that's been learned is if you wait too late, you may have to, in order to automate, you may have to repeat clinical trials. And it will take a bit longer to convince the agencies that you have a comparable product. So one of the things that we've learned, and we encourage our customers to really look at automation earlier in the process, is this not only gives you a stable process, but it also allows you to have conversations with regulatory agencies to show that you have locked down your process. And in a day and age where the regulatory environment is shifting, this becomes more and more important. At the end of the day, we all want to improve patient lives. And the regulatory agencies are right there. They want to help develop and accelerate the on-market delivery of these therapies. And so in doing so, they're trying to, give more access, give advice, and help different developers be able to bring these therapies to market. There's accelerated approval pathways. We see this globally. And the different regulatory agencies are harmonizing so that if you go down a pathway in the US, you can harmonize across the EU. And within Asia, much harmonization is happening between the different regulatory agencies, which is all great because it allows developers and researchers to get these products to market more quickly. However, this also creates a bit of a challenge because if you're able to get to market faster, that means the process development and whatever process you're going to be developing should be locked down a bit sooner. And so now the time period to not only develop your process, but to stabilize it has shortened. And with many different therapies that people usually have in pipeline, they will benefit from that lockdown. And so having that robust process, again, as I mentioned earlier, can help to accelerate that time to market. Some of the other things that we hear from our customers as far as some of the challenges that they face, one is manual methodologies. So many of these processes are manual. They start in a research lab. very academic-based, and they're trying to scale them. And what's challenging about a manual process isn't just the time that goes into it. It's that with cell and gene therapies, you already have a very complex process. You're changing or manipulating or expanding a biological phenomenon, and you're asking it to either do a different job or to do what it does in a more robust way. And when you have a manual process, you're adding additional complexity on top of that. There's also a need for flexibility, especially as we talk about process development. And so customers are looking for that flexibility early in process so that they can manage their process development as they manage the overall manufacturing. Another challenge is that you hear the phrase that the process is the product. So there's a lot of IP tied to the process, and therefore there's a hesitance to publish and to share learnings. And so this really limits how much we can learn from each other. There's also inconsistency in different materials, whether it's the starting or source material or various batches of product that come into the process. So you can see that this is also something that needs to be managed and controlled for. There's also variability in the different SOPs, protocols, standards that are used, whether it's an apheresis product that you're collecting and the protocol that's used there, or the various testing and release testing that's done along the way. There's a lot of variability there, and we see a call to action as far as standardization. There's also a need for more integration of data. So there isn't a shortage of data out there, but what's the meaningful data, and how can it be connected and shared across the process? I alluded to the standardization, and within the industry, you hear, I've heard it several times at this conference and several of the others I've been to over the past year or so, that the industry is calling for standardization. How can we standardize some of the basic processes just so that there's common terminology, if nothing else, but also to be able to understand, what are the data sets that are needed? And when you say standardization, it can be ambiguous. What are we talking about? But first we need to identify what are the parameters that need to be standardized? What type of data should be collected? And what's the impact of that to the final product that everyone is seeking to develop? What are we doing to help today? As an organization, one of the key things that we do, given our Optia collection platform for apheresis, is we're helping our customers be able to optimize protocols for their collection. We're also looking to develop and have products on the market that are flexible and modular so that you're not locked to a specific process. They can integrate depending on what it is that you need to do. And I see a call to action to the whole industry so that there can be a sharing of learnings across the industry and experience that everyone has accumulated as we've taken this path to commercialization. And really also partnering to help define some of the parameters as we look at standardization specifically and broader collaboration in the market. So as I alluded to with apheresis, we, with our Optia platform, have a very large footprint within academic centers, hospitals, and collection sites. And with that, we're able to really help our customers optimize their collection. And if you look at the cell and gene therapies today, about 60% of them that are in development, if you look at the clinical trials, they start with an apheresis product. And of course, we also know that there's variability from patient to patient in an autologous situation or donor to donor in an allogeneic one. And so optimizing this product will really put you into a better foothold as you go into your manufacturing process. And that's one of the things that we look to do. For this slide, I wanted to really show the continuum of care, and as you can see here, everything kind of starts and ends with a patient, whether it's an autologous product as is depicted here, but even in the case of an allogeneic, it starts and ends with a donor and then a patient. And we need to remember that and keep the patient in the forefront of our minds during the development process. And regardless of the type of product, there's several different steps and processes that are followed when someone is developing a therapy. And I'm not going to go through each of these steps, but I will say that, during the modification or the expansion phase is really where a lot of the heavy science happens. This is where you're modifying the cells, you're adapting them, you're changing them, or you're expanding them. And that's a very different level of effort. What also happens if you look at the downstream processing, where you have the cell washing and concentrating, or the fill and finish of the final formulation, in the downstream processing, that tends to be a bottleneck for many developers, because now you have this large volume of product and you want to be able to concisely concentrate and package it for delivery and shipment. And of course, time is always a variable here as well. So I wanted to share one of our platforms, and this is the Quantum. Our Quantum is a hollow fiber bioreactor technology. And really what's nice about the Quantum, and I won't go into all of the details here, but it is a very flexible platform that is able to do a lot of different things. So we have published pretty extensively, us and those of our customers that have used this system for adherent cells, but this platform is also used for suspension cultures as well. Exosome production, viral vector production. So a lot of flexibility in a single platform that's a functionally closed system that can be used in a number of different environments. And I won't go through all of these, but just to show a reference, and this is just a collection of some of the publications that have been done by some of our customers using this platform in this space. I'd also like to highlight the Finial fill and finish system, which was launched this year. And there was a press release that came out this week as well highlighting this. It's really the first device of its kind that is able to, in a fully automated, in a functionally closed manner, take your final cell product and mix it with so it can take up to 3 components, so your cell product, any other buffer, and the cryo, in order to cool, to mix, and to aliquot that material into a pre-specified volume that you would enter. And once it does that, it removes the air and seals it. So really these bags are ready for cryopreservation, which really helps to speed up this part of the process, but it also adds a layer of control in order to be able to manage the process more effectively, especially because there's oftentimes a concern for how long the cryo will sit with the cells given the impact on viability. And this system helps to control that. I'd also like to highlight that, as we bring products to market and help our customers, we also are very proud to be a collaborator of choice with many key leaders in this space. And so the irony is not lost on me that we are in the Cognate room, and Cognate is one of our collaborators, and we're really delighted to be working with such leaders in the industry as they do not only contract manufacturing, but also process development. And so there's a really great synergy between our organizations. Cell to Cure is a clinical stage organization in Europe, and they are also using some of our platforms in their development in cardiovascular disease and sharing these learnings with other investigators. And Unicar is a CAR T therapy company in China looking at the next generation of CARs coming to that marketplace. And finally, I would really just like to end the presentation to highlight some of the stars in our organization that are here this week, Lisa Williams, Rhonda Wallen, and Deidreanne Perry. So please do reach out to us if there's anything that we can do to help. We have a unique vantage point in the marketplace in that we've had the benefit and the privilege of working with many different companies and seeing different processes. And we've learned a lot along the way, and always love to engage with others and continue the learning, but also share that as well to see how we can help you bring tomorrow's therapies to market. So with that, I will end. Thank you very much.